And we'll talk about some of the factors that are associated with this progression. Now, what is the difference between a simple cold that somebody catches, which is an infectious disease and it can spread, and something that eventually becomes an epidemic? So epidemics are localized outbreaks that typically have a beginning and an end, and pandemics are similar outbreaks that go beyond some region, they go global. And so what we'll look at today are a couple of examples like that: Ebola virus and coronaviruses. One is an epidemic, another one is a pandemic. So we'll talk a little bit about some of the factors like viral adaptation between different hosts, the origins of viral outbreaks, virus transmission, cell entry, and tissue tropism. And then we'll talk about how the same factors at the micro level affect the disease progression stages and how those could be characterized. Before we jump into this, let's review some of the progress. Let me know how you are doing. I know that Dr. Manav, right? You had some questions you posted on the forum. Were you able to find the resources that we discussed last time?
Yeah, after that I was able to find out, but in the code omics I posted two technical issues also, like I was not able to proceed.
With what exactly?
In the code omics, I was just practicing R. So after like step 2, like I was not able to proceed. It was not, the code is showing error. So can you
Okay,
please look at
great.
it?
So yeah, so why don't we keep that for later today, and we'll talk about
Yeah,
some of
sure,
those
sure. No
technical
problem.
challenges with Harpreet. Okay, excellent, thank you. But I did want to just point out that here in the forum, I paste the links from the sessions that we discussed. They include some topics that we discussed that are linked in the papers, and we'll talk about quite a number of publications today as well. I understood from our last conversation about the pipeline outputs that a lot of you have questions that are conceptual. Why are we doing what we're doing, and what does it help us with potentially? And so I hope that some of these allow you to think critically about some of these challenges and how the methods are associated with that. You can see here that we also post some results from the analysis and some data sets that we used. So hopefully you're finding everything here, and I merged your question here with all of this. So if you have a specific question about some of the resources or data sets, please post it here. I did also mention a number of different types of software that you could be using to do different things, kind of downstream analysis. So please take a look at those. In terms of also progress, remember that we have a number of courses, projects, and assignments. And so you will go through these courses either in between the sessions or after we have our sessions. You will have some time to work on those. And it's important to continue and practice those, because I think that you really understand some of the applications of the methods in the context of a project. And so it's important to define what your project is, whether it's one of the projects that we're doing in the sessions, like Ebola, malaria, and other types of examples that we highlight, or develop something around a topic and a project of your own, which will also help you because you're starting to think about the context, the actual pathogen, the disease, etc. So please try to go through some of these. Mona, I would recommend for you to complete your profile. Ultimately, you would want your profile to have a full name that will go on your certificate. And also you want to maybe add some of the links so that others can connect with you as well. If you're going to be doing anything with your project, you want to join with other people. So please take a look at some of the other people that have completed their profile. You will see what they have put into their profile as well. So I see that there's some activity, which is great to know. And if you continue like this, I think it will really benefit all of us to do this as we practice, not only in the sessions, but also a part of that. So let's review briefly what we have covered in our last session. if you remember, we did a couple of practical hands-on sessions. So the first one was how to start different pipelines, and then the second one was on pipeline outputs. So what are some of the different pipelines that we did? We looked at pipelines for visualizing, and you can see here a different example. This is an example with Ebola virus, but last time what we did together was we analyzed some of the samples from malaria projects, and we compared two different projects. Ultimately, the goal was to find a specific mutation that is present in one sample, not present in another sample. But the basic pipeline looked like this. I'm showing you this so that you understand that the same exact type of analysis could be applied to a very different data set. So for example, in this case, you see a similar approach to differentiate between a coronavirus sample, and that coronavirus sample could be mapped on two different reference genomes. So for example, the RAG13, which is a genome from 2019 that was sequenced from bats, but seems to be very close to the genome of the coronavirus that later on was found in humans. And this is the first published reference genome for the human infected. And you can see that the same sample, so you can see here 797, 797, 578, 578. You can see how those samples are revealing a lot of differences from these reference genomes, right? So this will give you some sense of how to work with such files. These are SRA files. You can see that they generate a BAM or a SAM file as a result of mapping and alignment. And you can also get some statistics from the different types of genomes, right? So you can see by just looking at statistically how many reads are aligned to the reference genome, what you're expecting to see. The other type of analysis that we did, and here I just wanted to explain a very specific thing that we did last time. I don't know how many of you actually completed the pipeline and have looked at results, but we talked about how alignment could be done at the nucleotide level or at the amino acid level. And so let me just quickly show you so that it's fresh in our minds. What are some of the important outputs? So together, we started this pipeline together, which was, again, just what you see here, multiple sequence alignment, conversion to the amino acid positions, and then select codon position MSA. We used also a reference GB file, so a file from the gene bank. And that contains the triplicate codon references for amino acid changes as a result of nucleotides. And so if you open the download all files, you will see here, this is a very important file that we will continue looking at, which is a full table.tsv. In this table, what you will find, it's quite a large data set or file because we have a number of different samples. But what's important about this file is that it shows you the connection between nucleotide changes and the changes in amino acids. So let me just briefly show you this result. Where you go? Okay, there we are. Okay, so if you look at this file, you see here, you see here the nucleotide positions, and you see that many of them, right, have a deletion in the beginning, many of the references. Now, if you start scrolling down, you will see that these become trinucleotide codons converted to amino acids, right? So now, if you see the changes, you can compare changes at the nucleotide level, and you can compare the changes at the amino acid level, right? So you can select, for example, these two, and you can now look at only the changes between them, between the amino acid references and not the nucleotide references. And we're going to do some practice exercises with these files later, but it starts giving you a sense of how the different levels of information are connected together, from nucleotide genome sequence to an amino acid protein sequence, and eventually to what these proteins do. So what is their functional domains, and eventually what changes in the functional domains can result in biologically significant functional changes. So we'll talk about in our later sessions, but I just wanted to make sure that you are aware of this important output from this pipeline that will only happen if you use a reference gene bank file. So whenever you upload your files, you have to make sure that you have that with you. I think if you go to here and you go to begin, then in the references, okay, maybe it's not placed here, but it is something that we uploaded. And so just keep that in mind whenever you're doing that analysis. Okay, so if we look at this kind of analysis, the output from here could be of multiple different types. So here we looked at multiple sequence alignment. We can just do it for the reference sequences, or we can also use what's called SHORA, which is a haplotype analysis. So if you have your FASTQ files, you can analyze variations within each sample and determine their relationship. For example, in this Ebola virus, you see very defined two groups, and those two groups are from different locations and different infections in Liberia and Sierra Leone. You can do a similar analysis here. For example, we have an analysis between Gabon, Liberia, and UK, right? So we'll give you today, we'll talk a little bit more about how do these analyses help us answer specific questions about epidemics and pandemics. And also, we talked a little bit about the same type of analysis being available not only in the phylogenetics section, but also in the section called viral quasi-species, which we'll talk a lot more in future lectures as well. So here you can see an analysis of coronaviruses, how they separate between different groups. And importantly, we can add this information about groups if we actually create files as input that are organized by different types of sequences, by location, by type of virus, by other factors as well. And so that gives us this ability to quickly separate the results. And we looked at how to build a phylogenetic tree in the pipeline, but then visualize it in other softwares as well. So importantly, what we are going to look at more and more in future lectures is how can we translate this information into biologically relevant variation. And here you can see an example. This is the sequence navigator that we already looked at a couple of times. And here, this TSV file that I showed you earlier is what allows us to map the individual mutations in these bins onto the protein surface. And here you can see the variation of the protein surface colored on this particular PDB file of that protein. Now, similar type of analysis could be done on other software. And this software, the limitation of that software is typically going to be that you have to install it on your computer. And so the RAM and the CPU limitations of your own machine might make it a timely and time-consuming analysis that you have to do using this kind of software. But ultimately, it could be done. And it's important to understand how some of this software works, including what we will do in future sessions is how to map the specific variations at the level of proteins onto structures. And so we'll take a look at that in our future lesson, but we do need to be prepared for that. So if all of you can take a look at some of the software, I posted these different links on the forum. You will find it there if you can install some of these. And then also we'll be talking about Chimera, or Chimera X in this case, which is a software to visualize the protein variation and protein models. And also we'll look at how can we link the different functional domains and changes in those domains onto the protein level. But we'll do that in the future. I just wanted to make sure that you have prepared, that you are prepared by installing the software on your computer. Okay, so let's jump in what we are planning to talk about today. And I hope that any one of you that has any questions about the things that we are going to be talking, that you can actually put in your questions in the chat, and we can address those questions at the end. So the plan for today's session is to kind of give you a high-level overview of some of the important topics and questions and link those to a pandemic or an outbreak and some of the factors that are related to that. But if at any point in time you have a specific question or technically you have some challenge, please do let me know. Then at the end, we will do some hands-on activity, which will include using the logic from today's session to select specific file types and samples to make sure that you can translate the questions into an analysis. And you kind of can link the analysis part with the biological and the clinical significance of the analysis question. So before we move on, does anyone have any questions so far about what we talked about last time? Some specific things that you would like me to address before we move on? All right, great. So if you do, put it in the chat. So today we'll talk about two different categories of questions that could be asked using such genomic data. There are two kind of groups that we can separate them. One is the origin, or where did this infection, where did this virus come from? So that answers questions like, what is it? If we have a new infection, a new outbreak, how can we define what this outbreak, what is the pathogen that is causing this outbreak? If we can accurately characterize this pathogen, we can do a lot more because now we know who are we dealing with, right? Where did it come from also answers the question about potential reservoir, which is an important factor, and we'll talk about this in a couple of minutes, about knowing how to address the source and really identify whether that's going to be a continuous source of infection or is that something that was a one-time event. Geographic location is also important because if you think about it, you have a pandemic or you have an epidemic, people from one location are getting sick, they're moving around. How do we localize, how do we trace it back to its original location? And then how is it different from what we already know? If we had Ebola outbreaks in the 1970s, 1980s, 1990s, 2000s, how can we then separate them and know that these different outbreaks were caused by different types of viruses? How can we understand how are they related to each other and how are they different from each other? So we can then start asking more advanced questions that are more immediately important for an infection that is ongoing, an outbreak that is ongoing. So when we have the second type of questions, we are really asking questions about the relationship between genotype and phenotype. And that might be, how does it work? For example, how does this particular virus infect? How does it enter a specific type of a cell? How does it cause infection? And there we can characterize it by looking at some biological differences from well-characterized viruses that we already know a lot, because a lot of this information comes as a result of experiments done on cell lines in animal models, very detailed type of knockout experiments, as well as cryo-EM microscopy and other types of ways to characterize the protein and the metabolite and the non-coding RNA variation. And so when an outbreak happens, the questions about origins and comparative genomics that allow us to compare sequences between different types of strains that are well characterized can really advance and narrow down the important questions that we might have with association to a phenotype. Now, importantly, we can start asking questions that are coming from the tracking that we have data. For example, rates of transmission, mortality, speed of disease progression. All of these questions are immediately going to come from epidemiology and the clinic, and then we can try to link the biological considerations around sequence similarity to some of these phenotypic insights that we get from epidemiology and clinical data. Ultimately, an important question that we can start defining are going to be around the targets, so protein targets, targets of interference, and sensitivity, so sensitivity to different types of treatments or vaccines and other types of measures that could be implemented. So we'll talk about how all of these connect, but let's take a look at how can genomic data characterize infectious disease outbreaks and kind of what is the history behind all of this. So the impact and importance of genomics in pathogen characterization is routinely demonstrated in all of these different outbreaks that we see. And importantly, one of the factors is the rapid prediction of and response to, as well as mitigation of outbreaks that requires a lot of information about genomics of virus consensus genomes. And so when we have a new outbreak, we have this reference. So, and in the context of these different infections, you can see here in 2003, the first SARS and MERS were caused by coronaviruses, right? So in a sense, this novel coronavirus is not very new. We had a lot of reference sequences because this happened over a decade or maybe almost 2 decades ago. Then there was this H5N1 bird out flu, H5N1 and then H1N1. So both these came from birds and swine. Cholera, MERS, Ebola, Zika, right? So you can see that over time, a lot of these outbreaks have happened. And if you look at all of these, what do you think is a common theme that you can quickly see in all of these viruses? What kind of stands out to you as you look at this sequence of outbreaks? Is anyone looking at this chart and saying, okay, I see some pattern here? Okay, so what do you think, Mona? You can unmute yourself. Okay, so you're not sure. So what is a common pattern here? If you see, this is bird flu. Okay, so mostly in a particular region. So Ebola, you can see, so let's see, yellow fever maybe, right? Plague in Madagascar, Ebola again. So we see here swine, bird, Ebola, again, we know that is also coming from bats. Zika potentially also came from animals. Ebola, obviously, again. So a lot of these are specific to regions, right? So there's the geographic region. And a lot of them come from different types of animals, right? And a lot of them are caused by RNA viruses, right? So we can study these different trends. We can link them to other factors of what happened in those years, and we can start characterizing them. Now, an important asset, because we have genomic sequencing technologies that are widely available, you can see the impact of genomic sequences on these different outbreaks. For example, during SARS, this is an average number of full genomes per month that were submitted to NCBI viruses. And I think this was done in April. So you can see that in each month, right? So for the duration of SARS in 2002 to 2004, on average, there was about 3 genomes that were published on NCBI. For Ebola, there was a major effort put into metagenomic sequences. And that's why we are focusing this program on Ebola, because so much data is available and it was done as the epidemic was ongoing. So then some of these other also produced a lot of genomic data. And ultimately, during COVID, there was this exponential growth of genomic data that was made available about the virus. So we have a lot of genomic information. How can this genomic information be used? So targeted or unbiased sequencing of individual clinical samples aids in the identification of outbreaks, the determination of outbreak etiology, and the definition of virus transmission chains by identifying chain-defining single nucleotide polymorphisms, or SNPs. Also, field transcriptomics improves our understanding of host responses to virus infection and is important in deciphering the differences between asymptomatic and symptomatic disease states, and in predicting whether patients with acute and chronic disease will survive. Functional genomics is also becoming a tool for rapid characterization of patient-specific viruses that have not been isolated in culture and that cannot be equitably shared among labs across borders. And finally, genomic analysis of patient-specific viruses also enables precision medicine by predicting the efficacy of available medical countermeasures against these individual cases. So as we said, one of the emerging patterns here is that many of these diseases are caused by transmission between animals to humans. So about 60% of existing human infectious diseases are zoonotic, which means they came from a different animal host. And at least 75% of emerging infectious diseases, including Ebola, HIV, and influenza, have an animal origin. So 75%, the majority of diseases, cause important outbreaks because they come from different animal reservoirs. The transmission of an infectious agent from an animal to a human being initiates a series of events that constitute the pathogenesis of infection. Pathogenesis is the entry, primary replication, spread to target organs, and establishment of infection in the target organs. The process by which a pathogen replicates itself in the human host depends on cell-specific and organ-specific receptors, cells and tissue injury, and host immunity and other defense factors. The final outcome is either termination of infection, persistence and latency of infection, transmission to another host, or some combination of these. This series of events is not specific for zoonotic infections, except possibly that zoonotic agents are rarely sexually transmitted. The zoonoses, however, illustrate some of the more interesting and complex patterns that have evolved in nature. And so because of these differences between immune systems and the conditions of these hosts, many of them have conditions that are very acidic or very strongly influenced by their immune response. These viruses have the potential to really cause severe damage when they transmit to humans. And so this is one category of questions that could be addressed with genomic sequencing, is where did the virus come from? Which animal or which reservoir contains this kind of a virus that has an important potential to cause an outbreak? And some basic questions about what might be the disease by comparative genomics. Now, another important factor here, when it is already in a human population, is accurate characterization of the virus. And there are a lot of factors here to consider. So I know that you might be looking at this chart and thinking, okay, what are we trying to get out of this chart? This is a chart for clinicians to characterize viral hemorrhagic fever. An important part of Ebola is that it has a very complex pathophysiology that is characterized by immunosuppression as well as stimulation of an intense inflammatory response that results in syndrome similar to septic shock. And the diagnosis early on of this disease is difficult due to the initial symptoms that mimic other diseases. In fact, for about a week after a person has gotten Ebola, there are no symptoms. So the person feels fine. And then eventually when symptoms start to manifest, you observe a very similar fever, diarrhea, so very common diseases that might be the cause of that. And so obviously, many times the patients, when they arrive to a clinic, it is too late to do anything about it. And the treatment of the symptoms is the immediate factor that the clinicians would focus on. And so here you can see a very complex sequence of questions and tests that a patient would have to go through. And the ultimate goal here is to identify and contain the person that does have an Ebola virus to prevent transmission, but also to identify specifically what type of a virus is. And so here you can see an example I thought was very neat of a teenager that created an Ebola diagnostic test. The genomic PCR testing, to characterize the genomic kind of cause of the pathogen is difficult because it requires specific cooling for that to work well, and there's a delay. And so she created a test that does not require cooling and is fairly accurate, and so it could be easily deployed. But an important factor here, right, is early detection and accurate characterization. So accuracy and time are very important factors. Ultimately, you can see the measures that they went through to contain the disease, to prevent spread of the disease, which spreads through bodily fluids. And so an important factor about these types of questions that we can ask has a very practical clinical application that is not beyond any one of our ability to contribute to as well. In many cases, these types of events will continue in the future. This is not the last epidemic or pandemic that we are seeing. And it's important to think practically, how can our ability to work with genomic data help us to identify these emerging pathogens and help characterize and define and diagnose them early on in the outbreak. So let's talk about these in greater detail. And first, let's talk about zoonotic transmission. There's actually no clear understanding on whether emerging viruses become more or less virulent following a jump to a new host. Before a host jump, pathogens are likely to have been selected to optimize their virulence for onward transmission in the reservoir host, right? So if the evolutionary strategy of a virus is to replicate as many of its progeny as possible, killing the host is not favorable. In fact, what is favorable is transmission between hosts, because if the virus can be latent in many hosts in a whole population, it's going to reproduce much more effectively than if it just kills a small proportion of that population of hosts. But immediately after the host jump, the pathogen is likely to be initially poorly adapted to the new host, which may inhibit successful emergence, and the host is likely to be poorly adapted to the new pathogen. High host mortality is associated with a reduced likelihood of successful onward transmission in the case of human viruses, right? So if the virus causes death, the transmission is going to be something that humans are going to be trying to identify and stop right away. And there are three possible scenarios for virulence evolution immediately following a host jump. So here you can see some of them. Some of the most devastating epidemics have been associated with an increase in virulence following a cross-species transmission. So these include HIV, that almost invariably progresses to AIDS if untreated in humans, in African non-human primates, and the closely related simian immunodeficiency virus, CIV, persists without causing disease, right? So if you have SIV, or the disease where HIV came from, it is persistent. It does not cause major symptoms of disease. But once the host jump happened, it started causing death and AIDS. In another example, we can see no apparent change in virulence following a host jump that can be seen in H3N8 influenza A virus that jumped from horses to dogs. And disease is mild in both cases, and there are minimal biological differences between these viruses. Similarly, rabies virus causes an equally severe, ultimately fatal encephalitis in foxes and dogs, and following spillover to humans. Okay, so that means that no change in viruses. In one example, it's not causing death in both hosts. In another case, it's causing death in both hosts, right? So virulence does not change as a result of the host jump. And then in some cases, there's a decreased virulence that can be overlooked because avirulent viruses will often persist in a host population without detection. An example of a decreased virulence following a host jump may be that of the infectious hematopoietic necrosis virus, IHNV, that spread from sockeye salmon to rainbow trout in North America and Japan. And so sometimes there's a decrease in virulence. Of course, in human diseases, oftentimes we're not going to be aware of that because we're going to be focused on these types of cases. Ebola virus is a zoonotic disease, which means that it spreads between animals and humans. And the thought is that it came from fruit bats to humans. Once in humans, it easily spreads from person to person through contact with blood and other bodily fluids. Complicating containment efforts, the virus has a long incubation period. A person can be infected for up to 21 days before symptoms appear. Because of this long period of infectivity and poor sanitation in the area, the epidemic in 2014-2016 has presented a massive challenge to healthcare workers. And so what's important about this process of transmission is that typically there's some intermediate host, right? So the question is, fruit bats, when do we actually interact with fruit bats? Well, not a lot, but there is indirect contact. So if the virus from the host goes into an intermediate host where humans do interact with those, for example, eating the flesh, eating the meat, and contact with bodily fluids happens right there, then there's a high chance of transmission, and then you have all of these additional factors that characterize how fast the disease spreads. Viral DNA in Ebola viruses, which replicates in the cells of the infected person, lacks the proofreading mechanism present in humans and other organisms that detects and corrects mistakes made during DNA replication. The lack of this proofreading mechanism results in a high rate of DNA mutation, allowing the virus to evolve quickly, adapt to new environments, and elude potential treatments. So this is an important biological characteristic of these RNA viruses. They contain material that is not very consistent in general, highly variable already, and they also don't have this proofreading mechanism. As a result of that, it could adapt very easily. Genomic analysis of Ebola viruses reveals that potentially Ebola virus likely spread from Middle Africa within the past 10 years. And so they found that these viruses that causing the outbreak and two previous outbreaks diverged from a common ancestor around 2004. This means that these outbreaks arose from different jumps from animal reservoir to the human population. So if you see an outbreak that was caused by zoonotic transmission, like in the case of Ebola, and genomic analysis reveals that there were specific events, and those are different events coming from a population of animals, you might want to think about how likely this is to happen again, and what might be the preventive measures to try and address that. And so there's an interesting study that was done where they tried to link the different outbreaks. So you can see these are two different outbreaks with a reservoir for the potential hosts. So here you can see the total population that is living in areas of risk of zoonotic transmission. and you can also see the predicted distribution of zoonotic Ebola virus. So it's only one type of viruses that has a more likelihood to jump over, and it is present in a particular species of bats. And so here you can see how those two are correlated, right? So here you have this population, and this is the predicted area, right? Very closely associated. Now, for that to happen, there has to be a specific type of a condition for this to be even possible. So viruses undergo genetic change by several mechanisms, including point mutation and recombination. Recombination is a pervasive process that generates diversity in most viruses. It joins variants that arise independently within the same molecule, creating new opportunities for viruses to overcome selective pressures and to adapt to new environments and hosts. Recombination occurs when at least two viral genomes co-infect the same host cell and exchange genetic segments. Different types of viral recombination are recognized based on the structure of the crossover site. Homologous recombination occurs in the same site in both parental strains, while non-homologous or illegitimate recombination occurs at different sites of the genetic fragments involved, frequently originating aberrant structures. A particular type of recombination, known as shuffling or reassortment, occurs in viruses with segmented genomes, which can interchange complete genome segments, giving rise to new segment combinations. Illegitimate recombination is relatively infrequent in RNA viruses, but in DNA viruses can occur at much higher frequency than homologous recombination. In addition, the exchange of genetic material between viruses is usually non-reciprocal, meaning that recipient of a genome portion does not act as a donor of the replaced portion in the original source. So recombination is a widespread phenomenon in viruses and can have a major impact on their evolution. Recombination has been associated with the expansion of viral host ranges, so how one virus can affect multiple types of hosts, the emergence of new viruses, like we are seeing with the coronavirus or specific strains of flu, the alteration of transmission vector specificities, so vectors are mosquitoes, for example, that could pass the virus, increases in virulence and pathogenesis, modification of tissue tropism, for example, a virus that infects typically the gut can go into the lungs or into the central nervous system, the evasion of host immunity, when the immunity is recognizing the virus and all of a sudden that whole segment changes, right? It cannot recognize it anymore, and the evolution of resistance to antivirals. So these are some important aspects to remember. So 3 things. First of all, there are specific points of transmission. These transmission are major kind of long-term evolutionary events. They are a result of biological nature of the way viruses work and the way they infect different hosts. Number 2, we can look at specific point mutations and characterize where that came from and where it is going. And the third factor is that sometimes this is not a result of randomly occurring mutations or SNPs, it is a result of recombination. So now all of this together can be kind of pieced together as a whole picture of spread in a phylogenetic reconstruction that we already talked about a lot. And this reconstruction can help us address some of the questions that we are discussing now of where did it come from, how did it happen, what does it mean, and where is it going, right? What might be the potential? So here in next strain for Ebola, we can see how the dots, which are genomic samples, can be compared to each other by a series of connections in this phylogenetic tree, identify specific points of separation between the different clades, and then start asking the questions, well, what do we know about these current persisting viral sequences, and how can we link it back to where they are and what is happening at the phenotypic level with the patients that we observe? And there are 2 approaches here. This is a short-term evolution approach, which is typically experimental, right? So for example, we can take a sample from a number of patients and infect cell lines and see what happens at the short-term evolutionary level. And we can also look at comparative long-term evolution, so we can understand how the dominant sequences from the patients that we have start spreading in the population. Ultimately, the goal is to link this to biology. And we looked at last time how we can find models of important proteins in the Ebola virus in this case. And here we know that one of the important viral proteins is this glycoprotein that connects to the host receptor. So we can start asking questions about the biology of the sequence and how it affects the proteins that are ultimately going to be important for antiviral drug design and vaccine design. So to summarize, we have a genomic analysis pipeline where we can do sequence alignment, phylogenetic analysis, and then link it to population genetics. Those can answer how the zoonotic origin, what is the zoonotic origin, how did that happen? It can help us understand the transmission between humans, and it can also start giving us a sense of geographies in the case of a pandemic. And ultimately, you can also start thinking about how this affects the actual characteristics of the epidemic or the pandemic. Now, here you can see some additional methods that we have covered already, but we will continue to use even in today's session. And how do they link to the specific epidemiological data that is important for us to understand the link between genomics and the clinical data or phenotypic data that we might have. Ultimately, this is important because we can link it to the disease progression, which we'll talk more in detail about in our next session. And here we are focusing on the detection, right? So early on, it's important to detect and characterize what kind of a virus this is. Then to try to link it to intervention and post-intervention to effectively treat disease and to stop the transmission events between patients. So in the case of Ebola virus, I highly recommend looking at this publication, which is called The Evolution of Ebola Virus, Insights from the 2013-2016 Epidemic, that will address some of the questions that were very important during that time. And one of the important questions about this glycoprotein was, well, if the receptors are so different between bats and humans, how did the virus actually get into the human tissue, and what adaptation has happened since then, especially in this glycoprotein that is responsible for cell entry, because the potential was that there were specific mutations that were causing an increase in transmissibility and its pathogenesis, which ultimately led to death, as well as the potential of this virus to become airborne, which was something that people hypothesized about, but it never happened. Okay, so when we do this, we will actually talk about some specific types of analysis that could be done, including the reconstruction of the specific types of relationships that we will look at today. So first of all, I will put all of these different types of links in the forum for you to look through after the session. And importantly, there is an assignment for our discussion next time. So if you could spend some time to read through, and I'm just going to put this in chat right now as well, if you could take the time to read through this publication and answer some of these specific questions as you look through. So how many samples were analyzed, what two major lineages were found, which types of mutations helped characterize the two lineages, and what might be the role of a specific mutation in the surface glycoprotein that is described in this paper. Okay, any questions about this part of the session today? Does anyone have any questions about what we talked so far? Okay, so let's see how we can find the right kind of data, and I will pass it on to Harpreet here in a second to guide you through some hands-on practical activities. So let's go to NCBI virus. Okay, we've already done this, but I'm hoping that by now the reasons for the analysis and what type of analysis to do is starting to make a lot more sense. So one place we'll look at today is NCBI virus. Please let me know once you are here. Okay. So once you get here, you go to search by virus, and we will start by putting in Ebola right here. And this is Ebola virus. Okay, and you should see here over 3,000 nucleotide sequences. Now, important to remember that these are from different hosts, different locations, right? And you can see some of the hosts right here. You can see some of the locations here, the length of the sequence, and the release date, or also you have here the collection date, right? So these are the important areas of information. And what we would want to do in this first exercise is to ask the question, what might be the origin of this outbreak in 2014, 2016, based on sequence analysis? So please think about what sequences do you need to select from this database to be able to answer that question? And then what kind of analysis would you need to do to be able to start answering this question about the potential host? So please put your responses in the chat box, and Harpreet, I will pass it on to you to take it over from here.
Thank you, Ilya. Just give me a second to share my screen. So, hello everyone. So, how many of you are already on this site? Just let me know. So, on the NCBI virus. Okay, Dr. Manav, Mona. So, that's great. So, okay, if we are on this site, so first of all, let's learn how we can download our data set, especially if you're working on a viruses. So if you want to know what's its origin and where it has been started. So let's just, once you are on this site, so always we need a reference genome. So we can first select the complete sequence types. So here you have 6 reference sequences. So then you have sequence GenBank. So then based on even the country origin, geographical region, so we have multiple sequences from different regions. So let's try. So how you can start your problem. So you know that Ebola virus is spread in the Africa. So you know that from which region exactly it started. So how we can understand that. So let's select, first of all, first download a simple reference genome. So how you can find that what are the reference genome? So first you should just go to here, just select the reference genome, right? So once you selected that, so then you will find here different reference genomes. So it's like 2018, then 2010. So we are starting with the first reference genome, which was sequenced in the year 1999. So if you select this, so then you just go to the download option. So this is our reference sequence. So depending upon, because here we are interested specifically in the Z Ebola virus. So we should know, we should take that particular reference sequence. So there are different types of Ebola viruses. So the first Ebola viruses that was first sequence, that was Ye Ebola viruses. So to download this, just go to the download option. So here, so if you remember from your previous experience, so the reference genome is always a FASTA sequence. So then it will ask us in which format that we want to download. So just go to the next, then select it as we want to select only the selected record. So just go to next, and then it is asking which kind of definition line that you wanted. So we are taking accession number and generic title. So if you download, so this is our reference genome. So just let me know how many of you already able to download this reference genome. Okay, Mona, that's great. So now let me make a folder and move it to other folder. So this is how it looks. So I'm saving this as reference genome. So save it as reference genome. So then next questions that this is the reference genome once you have downloaded. So now we can unselect it. And if we are interested the other genomes, so just let's go back. So here I was interested. So let me just again search it. So here Ebola. So then you can filter it out here. So nucleotide completeness. So just select the complete sequences. So already you have done, so I just clicked it back. So then here you know that first of all, you want to select the sum of the sequence. So Dr. Manav, it is like should be always choose the earliest release, you know. So because we whenever a new outbreak happens, we want to know that whether it why we are taking the first release. So it is not always important, because even if a new outbreak happens, we don't know which category or which it belongs, right? So to understand we are taking that particular, we can always compare we can always generate a different reference genome, we can take all those. So I have just downloaded the reference genome first, Mona. So it is not important, so because whenever any outbreak happens, we don't know what's its origin. So we always take the first, we want to compare whether it is related to the first outbreaks or how many outbreaks that happens. We can generate a phylogenetic tree. So we can create the phylogenetic trees, and we should understand that with which it is associated. So you can take all the reference genome to understand your new outbreak is related to, or evolutionary related to, which type of outbreaks. So I have just taken the related to the GER. So what the next question that you have, what if the genome is not complete? So in that case, we can generate again, we can generate a reference, I mean, we can generate a phylogenetic tree. So if we have already the complete genome, so we prefer that. So in case if there is no complete genome, even if you have just derived a new species where you don't have, so in that case also you can take that. So if we have, we always prefer the complete sequence. So now if you look at this, so these are from different regions, right? So if you look at even the date of their release, so you just take, like if we want to understand it's their origin belong to which category. So I have already taken the, so let's start with the Liberia, that was recently, so 2019. So then we have a Democratic Republic. So let's select these three, and then we have 3 genomes from this country, that is Sierra Leone. So this is, we are selecting based on different reasons. So you can take 2 genomes and you want to understand from where it is get originated. So then even we can, so then we move to the next reference genome. So different countries that we have, we can select a particular genomes and we can generate a phylogenetic tree to understand where it is originated. So one of the way that with which we can understand from where it is generated, so that is the multiple sequence alignment eventually to generate a phylogenetic tree. So phylogenetic tree help you, so from which category it belongs, so where it is originated, so how we can understand that. So let us select few of the genomes. So let me share, I have already selected the 6 reference genome. So you can select all these reference genome that you want to compare in the complete sequences, and then we are comparing Sierra Leone. So this is from the year 2015. So we can also even consider even the new, we can understand, we can determine and analyze whether the recent outbreak it is actually associated with the previous one or not. So like here for the Gavin, this outbreak happened in the year 2013. So we can take this as well. So then even the outbreak that happens before that, so based on the even release date, we can also select that. So this was the recent outbreak. So we are just want to the last outbreak, that is in the year 2015. So Liberia. So then there was outbreak happens before that even. So we have taken these samples 14. We have selected 14. So you can select any one. I mean, you can select 10 genomes or 20 genomes. So depending upon the reason that you want to understand. So you can select the desired genome or genomes. So then you just go to the download. So here, if you want to know their phenotypes and which country it belongs. So first of all, let's download its nucleotide FASTA sequence. So the similar as we have downloaded the reference sequence. So beside, we can also download it is in the CSV format. So what's the advantage of the CSV format is that so that you will understand its genotype, phenotype, so you can select this kind of information. So like that you want to understand what's its host, what's its isolation source, so whether it's the geolocation, so then the genotype. So this kind of information you can select in the to understand that which phenotype it belongs. So you can also select this information and download this. So now let me show you. This is sorry. So this is what we have obtained. So first, this is just the phenotypic information. So we know that now we have 4 genomes for Liberia, so 3 genomes for Gabon, so 2 genomes for democratic countries, so Democratic Republic. So then their release date, even the years which with it released, it is like 2000. From the 2014 we have these 5 genomes, then 2015, then even we have the genomes from the Gabon, that is the initial outbreak that happened around in 1996. So we know that even from their ID, what is their phenotype and how they are associated with each other. So this information is important when you are analyzing, when you are finally going to analyze your phylogenetic tree. So then we have this sequence file. So I'm just and naming it as all the genome sequences. So I'm just naming it all, that is 14 genome sequences. So once we have these two files, one is the sequence that we have loaded and the reference sequence. So now let's go over our platform, that is Bioinfo platform. Just let me know how many of you able to download these two files. One is reference genome, the other one is the genome sequence that we want to analyze and eventually to understand their origin. So how many of you able to download these two files? Just type yes. If not, you can select any of these. I mean, you just select based on the year or the region. You can select any number of phylogenetic any genome to generate a phylogenetic tree. So it is here I have selected 14. So let me share the other link. So you can select even 9, 8. So depending upon your analysis that you eventually want to analyze. So let me just share one link from where you can download 9 sequences. Okay. So the sequences that I have loaded, so I have selected 14 sequences. So I'm pasting the link of these 14 sequences here. So just let me know how, just if you click on these 14 sequences, you will just direct it to all the selected sequence that I have loaded. So just let me know how many of you able to direct it to this page and able to get. Okay, so how you will understand that these sequences, how many sequences they have. So if you look at this selected results, here you will get that number. So once we selected this, then just go to the download. So here under the download option, here you will find if you want to download the FASTA files, FASTA sequences, just go to the nucleotide and go to the next, and then you can download directly. Then to obtain their phenotypic information, just select the CSV format, right? So because eventually once we able to generate a phylogenetic tree, so how they are related on exactly what information that they contain, so that is very important to understand. Even if you don't want to download your reference genome as individual file, you can also select reference genome from here as well. Sorry. So just let me know how many of you able to download FASTA file, FASTA sequence, so that we can proceed for hands-on. So any one of you able to download? So, okay, that's great, Mona. So apart from Mona, who is able to download these files? So even if you don't want to download these files individually, you can also select even the reference sequence at that point of time as well. The starting, even at the 15 sequences, so you can directly download 15 sequences as well. I think that will make even more easy for you. So even if I select this, so now I have obtained total 15 sequences, because we can perform we can generate a phylogenetic tree using all these sequences. We only know the ID, so the accession number. So even let me just, even if you are not able to download this, so let me just share one more link, so you can directly download these 15 sequences, and we can generate even a bit phylogenetic tree with this as well. So this is the other link. So where even if you don't, you have not downloaded. So here we have all 14 sequences plus the reference genome. In the second link. So just if you click on this link, you just go to the, once you click on this link, so just paste it. Sorry, okay. I think there is something missing. So let me just paste you the other link so that I can help you out. So, okay, select any 5 or 10 sequences that you want to analyze and you want to understand their phylogenetic relationship. So now let's go to the server. Okay, so I'm pasting one more link with you. So if you click on this, just click on this link that I have shared now. So here we have 9 sequences. So I think you should go with this, because if you are not able to download any of the reference genome, so just everyone, I have pasted a new link, and just click on this and you can directly direct it to this 9 nucleotide. Then just select it, select all, then just go to the download and then just proceed for analysis. So now let's go to the server. So does anyone download this 9 sequences? If you're not able to download, so if you are not able to, yes, okay, Dr. Manav. So just download these 9 sequences. If you're not able to download those 14 sequences, so I have created a new link, so with which you can download these sequences. Otherwise, you can select any of the sequences that you want to analyze, right? So once you are on this, not just, so once you're able to download both reference genome and these 9 sequences, so just next go to this Bioinfo server, and on Bioinfo server here under the virology, so if you click on virus cases, so just let me know how many of you able to direct it to Bioinfo server and are able to download this file. So here under the first we have to upload files. So here format of NGS data. So in these files we have the FASTA sequences. So from the format of the NGS we are selecting FASTA, and then we are going to upload here our file. So I am here now uploading the sequences that I have shared with you, that is 9 sequences. So here we have to upload 2 files, that is sequences and the reference sequences. The sequences is 9 sequences. So once we able to load this, then go to the group selection. So I am just creating 2 groups. One, I have just updating group all the samples in the group A and the reference in the group B. So then we can skip this. So then we just direct it to start. So once we able to start, we able to upload our file. So here I have only uploaded 2 files. So even if you want to create your multiple groups, like you are interested in the from the year from particular geographical region, right? So you can make select 3 groups, like 3 sequences as single file, and then again the 3 sequences from other file. So like in the here this 9 sequences that I have shared with you. So this is okay. So if you look at this, so from which countries it belongs. So if you look at the first, here we have 3 sequences from the Gabon and the year around 2013. Then we have 1, 2 and 3 sequences from the United Kingdom, and then we have 3 sequences from the Liberia. So we have chosen 3 different countries, and even based on the year we have 2013 sequences, then we have sequences from the year 2014, and the latest one from the 2016, 15 and 14 from the Liberia. So here we know that which character with the accession which kind of sequence that we have and from which country it belongs. So if on the Bioinfo server, next once you upload your file, that start, then just the next is multiple sequence alignment. It will align your sequence with each other. So once we able to done this, save this, then the next is because we have nucleotide sequences. So next, how we can convert this multiple sequence nucleotide into the amino acid. So the next step that is getting highlighted here, that is multiple sequence alignment, that is amino acids. So then the select position codon position. So if you are want to next, what options that you are getting? So codon third position. So if you look at here, when the third codon position chosen, MS is generated for sequence consisting only of the third position at each of the codon. So if you choosing the 4D position, then MS is generated for specific codons in which the change of the third nucleotide does not alter the amino acids. So this is very important. So if you changing the third position, so it is always considering. So if you choosing the fourth position, in that case their amino acids not going to change. So we are selecting the first third chosen as true. Then we are going to the save it. The next is there are various methods for the file to generate a phylogenetic tree. So here we are selecting the method that is beast specification birth death processes. So where it consider the birth rate and death rate of an organism. So here we are selecting that. So sequence type nucleotide, because we have here selected initially. So then we are going to end our pipeline. With this, we able to complete our pipeline. So this is our MSA pipeline for Ebola based on geographical reasons. So here we considering 3 geographical reasons. One is United Kingdom, the second is Gabon, and the third one is Liberia. So then just run pipeline. So how many of you able to run this pipeline? So how many of you able to run this pipeline? Just type yes. Okay, so just let me again just show you. So we have select from here, just let I can show you even in the pipeline. So if you go to this pipeline, it will take a bit time. Meanwhile, I can show you the results because I have already. Okay, so here first what we are doing. First, we have uploaded our files where we have sequences from different geographical regions. Then once we done that, then we have started the pipeline. So then the option that was getting highlighted, that is multiple sequence alignment, MSA. So once this is our nucleotide sequences, it aligns multiple sequence alignment, then it is converting into the amino acid, then we choosing the codons position. So here we have chosen the third codon. So if there is change in the third position codon, I think every one of you are familiar with that. In the codon we have 3 nucleotides. So if we change the third nucleotide, we know that amino acid can change. So if you check the fourth one, in that case amino acid will won't change. So then we check the phylogenetic tree method selected, that is beast speciation birth death process. So this method actually consider birth rate and the death rate of an organism. So then we just completed it. It is clear to you, Mona? Okay, that's great. So if you're able to run this pipeline, so now let me just show you the results of this pipeline. I think it will take a bit time to complete. I have already run this pipeline before. I had asked because it is taking a 16%. So once your pipeline will complete, here you will get the cluster. This is cluster W. Then you have this that process PDF. So this PDF actually contain the phylogenetic tree. So if I download this tree, so just let me show you what it representing. So here, if you look at this, so how you analyze this phylogenetic tree? This was our reference genome. So now we can, if you look at this data set, so if you look at these results, so what you will actually find out, so if we look at, these are very closely related to each other, and this group is altogether different, right? So this group and this particular sample again, so once you have these samples, so you will understand that there are the 6 samples that are actually correlated to each other or evolutionary close to each other. So then we have the another group of samples that is actually different. So then you can analyze which country it belongs, right? So then it is, we have a reference genome. So what this phylogenetic tree represent? So which was the first where it start originated? So if you know that what it represent, so these clades, these connection, what it represent? So it represent the time. So from where it is first originated. So longer the, even if you look at this, so here you know that this particular, once you have this reference genome, so this timeline, this, it will represent the timeline. So as these genomes must be at later on they evolved, while if this timeline is small, then it represents that these samples are actually originated first. So this is horizontal line. These horizontal line will represent the time. So from the origin, if this is the, we consider it is origin at the start from where it actually this dendrogram forms. So if you look at each of these clades, so these uh, 2 clades are actually that are get originated later. So the first one, our reference genome. So if you look at this in comparison to this origin, so origin is this, and then this is our the first reference genome where we have sequenced the first reference genome, that was in the year 1999. So then there are some of the second, these few samples are actually originated near to this year. So if you look at this these sample, you understand that these were the samples that were actually we have selected during the that happened in the given countries, that is, and these were samples taken from the outbreak that was happened in the year 1999. So these were the latest samples, that is either 2013 and 2018 outbreaks. So I think if your pipeline, so how many, I think it will take a bit time. How many of you have completed this pipeline? So even after this session, you will once you have, and we have also downloaded this particular same 16%. Okay. So then if now let me just download this particular file. So we will understand exactly when these sequences they belong. So in that case, I have selected the 12 samples in my previous pipeline. So now let's look at, sorry. So I want to create the CSV file for these 9 sequences. So geolocation that we want to know, and then the host, and then the isolation source and collection date. So let me just download this. Now let's see where these sequences belong. Samples belong to which country, and what was their origin. So we have these IDs. So we know in this file we have these IDs. Now if you compare, let me just make it small. And we have this result that we had. So this was my reference genome ID. So if you remember your reference genome ID, accession number was NC. So you can always mark. So this was our reference genome. So it was originated in the year 1999. So that we have even checked at that point of time. Now look at these samples. So I have this ID that is KC2242798. So look at this group. So KC4. So this was my, so let me just paste, consider it here. So which country it belongs from, here we will understand. So just let me show you these three columns. So here I have shown you this sample 798, 797, 795. So if you look at this, these are the three samples. So first of all, they were taken from the 2013 year, and the country that it belongs, that is Gabon. So then we have these samples. So here we have 138. Sorry, just let me it. Okay. So if we look at these samples, so now we have this KP 204932. So here I have this particular sample. So if you look at this sample, it is from the Liberia. So then we have the samples that is KR, that is KR 025228. So this sample is this one, and it is from the United Kingdom. So if you look at all these samples, they are either belong to the Liberia, either UK, United Kingdom. So I'm highlighting it with different color. So we know that these samples are actually close to each other. So these samples that are originated previously, so this was the these were the original, that is this is what our reference, then we have the year that was this was 2000, around 2013. So then we have these samples that are actually belong to these particular countries, that is Liberia and the UK. So these are more close to each other. So if you get any sample, suppose you want to know that from where it is getting actually originated. So from this we clearly can observe that the epidemic that happens in the United Kingdom, that were actually originated from the either from this Liberia. So even so from this you can understand from with which it belongs to, or with which more closely associated with which type of the samples. So you will always, from the phylogenetic and phylogenetic tree, you can analyze all these things. So its origin and with which it is more closely associated. So this outbreak that happens in the United Kingdom 2019, so it was more closely associated with the Liberia. So it is not that gammon. So that from this we can conclude that its origin was, even the recent outbreak, the origin was it from Liberia or the United Kingdom, right? So is it clear to you how we can analyze or find out the origin? So with which, so once you have these results, so you will get this kind of phylogenetic tree. So even you understand that from even the timeline, you just understand that which was the first outbreak that happens. So if this time this horizontal line is small, it indicated. So if you look at here, so if this horizontal line is small, that it means it was the small period of time. So it is the longer period of time. It means it was the early outbreak, and it was the longer the this timeline, it means it indicated it was the late outbreak. So it happened in the more recent times. So, and from here we know that it is the first of our reference, then this outbreak actually happened early in comparison to these samples. Even from this simple phylogenetic tree will give you this kind of information. It's which was the early or the late, and what was the origin of recent outbreak. So we can always compare all these kind of from the sequence alignments. So then you can, apart from here, I have just taken the geolocation. So based on the country from which it get originated, apart from, you can always select the host as well. So here we have the host option, so we can select it if you are interested in the Homo sapiens. So if there are other sources as well. So like if you have the host from the bat, so we suppose we want to understand based on the organism. So the recent outbreak associated with which kind of, or with which organism it was more closely associated. So we can compare the host and generate a phylogenetic tree. So we can take we can always take all those kind of things. So here, as this was not here, if you look at here, so based on the host, how many hosts this particular infection that have, you can always select your host as well. So it is clear to every one of you. So now you can analyze, at least my pipeline get completed. I hope your pipeline will also complete it. So if I just look at this. So now if you look at this, here we have the first reference, then we have these organisms, so which it belongs to. So it is, as we have seen, this must be the Gabon, from the Gabon geographical regions. Then we have these Liberia and United Kingdom. So, okay, so you can also, that's great, Manav. So you can analyze and understand which actually, that's why I have just also downloaded that CSV file, because the CSV file will allow us to understand exactly with just using the accession number, we know that from which country it belongs. Beside, you can also select, whenever you're selecting, suppose you are selecting samples from particular country, you make a reference, makes the groups, like, so here then you can create group 1, group 2, group 3. So depending upon, you can always play with that. So when we here here, as we were selecting, let me just show you here, how you can prepare your multiple sequence file. So suppose we have I'm just want to show you, if you have multiple, suppose I have these 4 sequences file. So I just want to make them a different groups, so that it would be easy for me to analyze. So when you go to the group selection, so you can always create your multiple groups. So here from 1, 2, 3. So suppose I just want to set my, suppose I have just taken the 3 samples and make a downloaded it as a single file. So then I can transfer the second file, then I can select my third file. So as I can select my fourth file, and then create the group. So then here, as you look at this, here you will get those groups like group 1, group 2, group 3. So you know that which your sample belong to which group. So you can also always design all these things. So if you want to analyze for particular group 3 samples, you can separately download all those groups if you want to compare that. So it is clear to every one of you? Okay, that's great. Dr. Manav, apart from Dr. Manav, whose pipeline get completed? So now you can take any sequence and you can generate your own pipeline to make it more clear to you, to understand how you can ask your questions and how you can address all those questions using simple multiple sequence alignment and based on the this phylogenetic tree. You can select your desired samples that you want to analyze. So I think that's all for today's session. So thank you everyone. So you apart from Manav, anyone else whose pipeline get completed? So I think so this is very important to analyze this phylogenetic tree to understand their origin and any outbreak that happens. So these phylogenetic tree will always help us to understand based on the sequence only. If we have the sequences, we can analyze all these relationship, evolutionary relationship, and we understand their phylogenetic relationship. Okay, so thank you everyone. So who have not able to complete this pipeline, you can do it in the coming weekend. So you can try this out.
Dr. Harpreet, you can end the session.
Okay, thank you.